All right, biology students, today we are reviewing for our state end of course assessment, and we are going to cover in this review all things that we've talked about with regards to biology this year. For this particular session, though, we're going to only focus on cells. So we're going all the way back to the beginning of the year where we talked about life being organized from the smallest unit of life, which is the cell, to the great big biosphere. These are the focuses for today's review lesson. So if you'll remember, organelles and structures within a cell interact as a system to maintain homeostasis. We know that cells reproduce to maintain genetic continuity. And then we're going to talk about the structures of macromolecules. Again, we're going all the way back to the beginning of the year. And the structures with macromolecules within the cell are related to their interactions and carrying out all the cell processes. We're also going to talk about cell transport and how that helps the cell system to maintain homeostasis. And then we're going to end with matter and energy and how those things move through the cell. So if you'll remember when we talked about cells this year, we said there's two types of cells. We have prokaryotes and eukaryotes. Prokaryotes are single-celled organisms. They don't have membrane-bound organelles like our eukaryotes do. And one of the big differences between prokaryotes and eukaryotes is that prokaryotes lack a nucleus. So I told you earlier in the year to help you remember that, remember pro rhymes with no. So our prokaryotes have no nucleus. They include our bacteria and archaea. We also have eukaryotes. So our eukaryotic cells can be single-celled. They can also be multicellular organisms. They do have cells that contain membrane-bound organelles. And of course, our eukaryotes are going to contain a nucleus within the cell. And you can remember that because U rhymes with do. So eukaryotes do have a nucleus. This includes our plants and our animals, fungi like mushrooms, and then protists like amoebas. Now, each cell has a cell membrane, and that cell membrane is just going to serve as a boundary between the outside of the cell and the inside of the cell. If you'll remember, the cell membrane is very flexible and fluid, and the purpose in that is to allow the cell to maintain its shape, but also allow ions and gases and things that are important to the functioning of the cell to pass through the cell membrane. The cell membrane controls the movement of materials moving in and out of the cell. And so we have an adjective that we use to describe the cell membrane, and that's selectively permeable. So selectively permeable just means that the cell membrane gets to decide what comes in and what leaves the cell. If you'll remember, the cell membrane is made up of these things called phospholipids, and they form a double layer. So we call it a phospholipid bilayer. But in just a little bit, we're going to review macromolecules. And remember, one of the macromolecules are lipids, and our phospholipid bilayer is comprised of lipids. Another organelle that might be found in plants and fungi and some bacteria and some protists is a cell wall. And so a cell wall is a little different than the cell membrane. It's going to be a little thicker. It's not flexible like the cell membrane is. And the reason for that is because the cell wall's purpose is to protect the cell and then provide shape for the cell. There are other important organelles that you need to make sure that you know. The nucleus, of course, contains DNA, and this is what controls cell functions. We have the cytoplasm, and this is where our chemical reactions are going to take place. Again, cell membrane just controls what enters and exits the cell. We also have chloroplast and mitochondria that we'll talk about later in this lesson when we get to the energetics part of the review. But chloroplast, this is the site of photosynthesis. This is where solar energy is captured and then converted into glucose and oxygen. And then the mitochondria is the site of cellular respiration. This is going to produce our ATP, which if you'll remember is energy. Ribosomes are going to synthesize proteins. Vacuoles store water and waste. The endoplasmic reticulum is going to produce lipids, and this is also the site of protein synthesis. The Golgi bodies sort and transport proteins and lipids, and then our lysosomes are going to break down old, worn-out parts of the cell, waste, and enzymes, includes enzymes. Now, we said this earlier, but this is so important to understanding the whole realm of biology. But cell structures and organelles interact as an entire system. And the purpose in this is to maintain homeostasis, which is super important for all living organisms. So living cells just maintain a balance between materials that enter and leave the cell, and cell transport, which we talked about in our cells unit this year, plays a super important role in helping to maintain that homeostasis. Now, if you'll remember, there's two types of cell transport. We have passive transport, which no energy is required for passive transport. This is going to move with the concentration gradient, and there's three types of passive transport. We have just simple diffusion, we have facilitated diffusion, and then we have osmosis. We also have active transport, but now in this type of transport, energy is going to be needed because in this type of transport, we have molecules that are moving against the concentration gradient. And so we have bulk transport, which includes endocytosis and exocytosis, and then we also have protein pumps. Passive transport, again, is the movement of materials across the cell membrane, but we're not going to need any energy for this to happen. We're moving from an area of high concentration to low concentration. So I want you to think about a ball rolling down a hill. No energy is needed for that. Once that ball gets to rolling from high to low, as it moves from high to low, it's going to just sort of happen passively. So this is what happens with passive transport. I am going to go through the three types of passive transport. So again, we're going to talk about simple diffusion, facilitated diffusion, and then osmosis. So in simple diffusion, this is molecules are going to move from high concentration to low concentration. We can see this in the picture. So you have more molecules here. They're going to pass through the cell membrane to the other side, where there's a lower concentration of molecules. Since this is moving with the concentration gradient, from high to low, no energy is going to be required in simple diffusion. Remember, there are specific types of molecules that passively pass across the cell membrane in terms of simple diffusion. And this is going to be your small molecules, the ones that are hydrophobic, non-polar, not charged. And so examples of this would be like oxygen and carbon dioxide. Now, in facilitated diffusion, if you facilitate somebody, you help them. So in facilitated diffusion, we're going to get the help of proteins. And so these proteins are going to help these molecules move across the cell membrane. There's two types of proteins that might be present during facilitated diffusion, and that's channel proteins, and we can also have carrier proteins. But a protein is required in facilitated diffusion. That's how facilitated diffusion is different from just regular simple diffusion. We're still moving from an area of high concentration to low concentration. So we are moving with the concentration gradient, and so no energy is going to be required. Same thing with osmosis. So osmosis is a form of diffusion, but we're specifically focusing on water with osmosis. So that's the key. When you're taking these end of course assessments, if we are talking about the movement of water molecules across the cell membrane, we are talking about osmosis. So with osmosis, again, we're moving with the concentration gradient from an area of high concentration to low concentration, and so no energy is going to be required here. Now, there are three types of environments that cells can find themselves in. So let's look at these examples here. So here we have a beaker, and there are particles within the solution inside the beaker, and then the gray circle for each of these pictures is going to represent a cell. So I want you to remember solutes suck. So if you can remember that, in our picture, solutes are represented with the blue square. And so let's look at this first beaker. So in this first beaker, I want you to notice that outside the cell, there are more solute particles than there are inside the cell. And so if you'll remember, solutes suck. Solutes always suck water in their direction. So these solutes are like dehydrators. They're going to pull water in their direction. So since there's more solutes outside the cell, water is going to be pulled from the inside of the cell to the outside. And so what this is going to do is it's going to dehydrate the cell. And so the cell will shrink, it'll shrivel, it'll decrease in size. We call this a hypertonic solution. And remember, this year we said hyper, high. the cell will dry. So you need to know how water is going to move in these situations, and you also need to know what's going to happen to the cell as a result. So in a hypertonic solution, like we see here, this cell is going to dry, shrink, shrivel, decrease in size because it's losing water. Now, if you look at the second beaker, we have more solute particles inside the cell than we do outside the cell. So if you'll remember, solutes suck. They're going to suck water from the outside of the cell to the inside of the cell. What this is going to do is it's going to make the cell take on water. The cell is going to increase its size. It's going to expand. It's going to grow. And so this is a hypotonic solution. And this year we learned hypo, low, the cell will grow. Now in our third beaker, we have the same number of solute particles outside the cell as we do inside the cell. So this is called an isotonic solution, and water will move, but it's going to move equally in and out of the cell. All right, so that's passive transport. Now with active transport, again, we're moving against the concentration gradient here, so it is going to require energy because we are moving from an area of low concentration this time to high concentration. That's not natural, and so we're going to have energy to make that happen. On your end of course assessment, usually you will see like this little starburst with the letters ATP on the inside, like you see in this third picture here, for images and graphics that are active transport examples. So if you see that ATP, that is just a clue that it's requiring energy. This process is requiring energy, and it's probably a form of active transport. So there's three types of active transport that you need to know. We have endocytosis, which is the movement of particles from low concentration to high concentration against the concentration gradient, and we're moving particles into the cell. So you can see in this picture, this particle's moving into the cell, and so this is endocytosis. Remember, your prefix endo means into. Now, exocytosis is also the movement of particles from low concentration to high concentration. So we're still moving against the concentration gradient. Energy is still going to be required. But in this example, we are pushing waste outside of the cell, or particles outside of the cell. So when we're moving particles out of the cell, this is exocytosis. So the prefix exo just means out. You can also have protein pumps, which again is the movement of particles from low concentration to high concentration, and we're just going to pump ions across the cell membrane. A great example of this is the sodium-potassium pump, and oftentimes that will be an example that shows up on these end-of-course assessments. All right, now we're going all the way back to the beginning of the year, and we're going to focus for a second on those macronutrients, macromolecules that we talked about. Now, just remember, macromolecules are the foundations for the structure and function of every living cell in every organism. They are super important because they're the building materials of the body. They're also the storehouse for energy for all the activities that these organisms do. Most of the macromolecules are made of monomers, and then monomers bond together to form polymers. So we're going to start with carbohydrates. Remember, carbohydrates are a molecule made of carbon, hydrogen, and oxygen. You can see that in this picture here. This is the structure of one of the carbohydrates. The monomer of a carbohydrate is a monosaccharide, and there's some examples that I posted too: glucose, sucrose, glycogen, cellulose. Now for carbohydrates, you need to know that they are broken down for usable energy in the cell. This is a quick energy source, and there are 4 calories per gram for carbohydrates. So it's not going to provide a ton of energy, but it is an energy source for the cell, and it is a quick energy source. Now, specifically in plants, the carbohydrate cellulose is used for structural support in making cell walls, and sometimes that ties into some test questions that they ask. So I wanted to just remind you about cellulose and how that is an example of a carbohydrate that is found in the cell wall in cells. We also have lipids. So lipids, just like carbohydrates, are made of carbon, hydrogen, and oxygen. And just a reminder, do y'all remember when we covered macromolecules, I told you to remember CHO, CHO, CHON, CHOMP. And that CHO is the organic compounds or the components of lipids and carbohydrates. And then CHON is going to be our proteins, and then CHOMP is going to be our nucleic acids. We'll get there in just a second. So our lipids contain carbon, hydrogen, and oxygen. Lipids are very important for the proper functioning of organisms. We commonly call lipids fats or oils. They are insoluble in water. This is a characteristic that's really important, distinguishes lipids from other macromolecules. And then I put a structure here of a lipid, so you can see these fatty acid tails here. This is a triglyceride. And lipids contain 9 calories per gram. Let me go back. Carbohydrates contain 4 calories per gram of energy, and then lipids contain 9. So while this is not a fast energy source, this is a long-term energy source. It does provide more energy per gram. Now, again, lipids are the main component of cell membranes, and they make the cell membrane one of its characteristics is how it has the hydrophilic head and the hydrophobic tails that we talked about this year, and how that's important to the way the cell membrane works. And just remember that that is because the phospholipid bilayer is made of lipids. We also have proteins. So remember, CHO. So proteins are made of carbon, hydrogen, oxygen, and nitrogen. These are large, complex polymers essential for all life. And the monomer for proteins is amino acids. So amino acids join together to form those polypeptide chains that make up proteins. Proteins have so many functions within a living organism, but they're super important for muscle contraction, transporting oxygen to the blood, and then also immune health. Proteins include collagen, enzymes, hemoglobin, insulin, antibodies. Proteins also provide the cell with energy. So just like carbohydrates, it contains 4 calories per gram. And I do want to point out, while we're talking about enzymes, it's a good time to review just a little bit about enzymes that you need to make sure that you know. So remember, enzymes are biological catalysts, and their purpose is to speed up chemical reactions. They do this by lowering the energy needed for the chemical reaction to start. So we call that activation energy. But substrates bind to enzymes at the active site, which is very important. So the shape of an enzyme is important to its function. And there's some things that can cause the shape of the enzyme to be destroyed. And that could be heat, temperature, pH, also concentration of enzymes or concentration of the substrates. Finally, our last not carbohydrate, macromolecule is nucleic acids. We're going to spend more time talking about this when we review genetics, but just a reminder, nucleic acids are complex macromolecules that store and transmit genetic information in cells. So this includes both DNA and RNA. If you look at the picture, nucleic acids are comprised of nucleotides. So we have our sugar, we have our phosphate, and then we have our nitrogenous bases. And again, we'll review that when we get to genetics. All right, we know that cells use energy. They use energy for many different things, but they use that energy to make new molecules, including enzymes that we talked about earlier, and also to build cell organelles and membranes. They use that energy to maintain homeostasis. And if you'll remember, this energy is known as ATP, adenosine triphosphate. So we have the structure here, adenosine triphosphate, because we have these 3 phosphates here. And just make sure that you know adenosine triphosphate can be abbreviated ATP. And so this symbol here, this little starburst with the 3 letters, it shows up in a lot of diagrams for biology. So I just want you to know that that represents energy in the cell. So cells are important for trapping energy. We call that photosynthesis, and then using that energy. So we call that cellular respiration. And so we have sort of an image here. You need to make sure you know how photosynthesis, the outputs of photosynthesis become the inputs of cellular respiration. And then the outputs of cellular respiration become the inputs of photosynthesis. So it sort of works in a circular manner here. So it requires solar energy or light energy. Photosynthesis requires solar energy and water and carbon dioxide. And those are the reactants. And then at the end of photosynthesis, the products are glucose and oxygen. And then glucose and oxygen become the reactants for this chemical process known as cellular respiration. It's how our cells generate ATP. And then the outputs for cellular respiration, again, are going to become the inputs for photosynthesis. So what is photosynthesis? If you'll remember, autotrophs make their own energy, make their own food, and they do this through photosynthesis. They use energy from the sun to build carbohydrates in the form of glucose. And the key to this process is a pigment that is contained within the chloroplast of the cell. And that pigment, chlorophyll, helps capture that sunlight and generate photosynthesis. You need to make sure that you memorize the chemical equation for photosynthesis. There's no way around this. You just got to buckle down and make sure that you know it. So we're going to have carbon dioxide, we're going to have water, and we're going to have solar energy or light energy. These are our three reactants, the three things needed to get photosynthesis started. And then remember, this arrow represents our yield sign. This is like an equal sign. And then our products are always on the right of that equal sign or that yield sign. And so in photosynthesis, our products are sugar, glucose, and oxygen, which is O2. Now, the process of photosynthesis is quite complicated, so I just want to take the time to review some of the things that we've talked about this year. So we talked about the two main types of reactions that happen within photosynthesis. We have the light-dependent reactions where we split water molecules, and then the Calvin cycle. And so I'll show you an image of that on the next slide. All right, so let's review. Here we have the chloroplast. So when you think photosynthesis, think of chloroplast. And again, we have two reactants here that are going to be important to the first part of photosynthesis, which are the light-dependent reactions. The light-dependent reactions are going to happen in the thylakoids. And so this is where we're going to take in light energy and water. And then the product, what's going to be produced, is oxygen or O2 gas. And then we have some energy that's generated, you see here, that's going to power the next step of photosynthesis, which is the light-independent reactions. These reactions are going to happen in the stroma. And this is where we're going to take carbon dioxide, and then we're going to generate glucose. Now, once that light energy is used to make glucose or carbohydrates, any organism can then use the carbohydrates for energy. A lot of times students think that only animals go through cellular respiration, and that's not true at all, which is why I put these pictures at the bottom of your screen here. So organisms get energy from carbohydrates through the process of cellular respiration, and they generate ATP or energy from that. So they use energy to make energy. So what goes through cellular respiration? Well, plants go through cellular respiration. Animals go through cellular respiration. Fungi and protists go through cellular respiration, and then also some bacteria. Here's the chemical equation for cellular respiration. Just like with photosynthesis, you just got to memorize it. The good news is, if you can remember one of these chemical formulas, you can just flip it around because, again, the reactants of one are the products of another. So here in cellular respiration, we're going to use glucose. We're going to breathe in oxygen. I'm thinking about humans. We breathe in oxygen, we eat carbohydrates, and then that's going to create that energy or that ATP. And then some byproducts here are going to be carbon dioxide gas and water. There's three types of reactions in cellular respiration. We start with glycolysis, and then we move into the Krebs cycle, and then into the ETC or the electron transport chain. And so if you've forgotten about this, here's a diagram. Again, we start with glucose, and in the first step of cellular respiration, we have glycolysis. Glycolysis is going to produce energy in and of itself. Not very much, just 2 ATPs. We also have the Krebs cycle, which is step 2 in cellular respiration, and we're going to produce carbon dioxide here. This also generates some energy as well, not very much, but it does generate some energy on its own. And then the electrons produced during this step are going to go on to power the ETC, or the electron transport chain. And this is where we pull in oxygen. So think about you breathe in oxygen. So this is where that oxygen becomes important, because when you breathe that oxygen into your cells can then use it in the electron transport chain to generate 32 to 34 ATPs. That's quite a bit of energy. And then also a waste product or a byproduct is water. Now, cells use some of the energy produced during cellular respiration for cell division. And there's 2 ways that cells can divide. We talked about both of these this year. So we have mitosis, and then we also have meiosis, or meiosis, as some people pronounce it. Cells reproduce to maintain the genetic continuity of an organism, and we have said genetic continuity so many times this year. I feel like you should know what it is, but just in the event that you don't, genetic continuity just explains how traits are passed from parent to offspring. So let's go over mitosis first. So if you'll remember, mitosis. So think about your toes when you think about the process of mitosis. This is how your body cells or your somatic cells are going to reproduce. Mitosis is a form of asexual reproduction, and in prokaryotic cells, this is known as binary fission. And in mitosis, we're going to start with a parent cell. It's going to divide into 2 daughter cells. And what is super important about these daughter cells is they are genetically identical to the parent cell. Here's the process of mitosis. So you'll see in interphase, you have G1, S, and G2 phase. Just FYI, it is very important for you to remember that during the S phase, the synthesis phase of mitosis, this is where DNA replication happens. So we're going to duplicate our chromosomes in the S phase of interphase. Then we go into the mitotic phase, which includes PMAT. That's prophase, metaphase, anaphase, telophase. And then at the end of telophase, the cell splits into 2 daughter cells, and this step is called cytokinesis. If you're more of a picture person and you need like all the steps laid out in picture form, here you go. So again, you have interphase, and then you have prophase, metaphase, anaphase, telophase. This is PMAT. This is part of your mitotic phase. So this is mitosis. You can see the chromosomes have duplicated. The nuclear membrane dissolves in prophase. And then your chromosomes are going to line up in the middle of the cell during metaphase. Then they're going to be pulled apart. The sister chromatids are going to be pulled apart during anaphase. And then you're going to have this cleavage furrow that forms, and your cells are going to pinch off and become 2 daughter cells. And these daughter cells are going to be genetically identical to the parent cell. During meiosis, there are some steps that are very similar, but there's also some differences too. So remember, mitosis is how your body cells reproduce. This is how your cells repair themselves and grow, how organisms grow. Now, meiosis specifically relates to gametes or sex cells. This is how our sex cells are made. So mitosis and meiosis, different. So if you'll think about meiosis, some people say meiosis, which would make more sense here, because I want you to remember meiosis. This is how I am made. This is a form of sexual reproduction. So this is how our eggs and sperm are made, our gametes are made. And the end result of meiosis is going to be 4 daughter cells. So with mitosis, there were 2 daughter cells made. Meiosis, we have 4 daughter cells that are made. And it's very important for you to remember that these daughter cells are going to be genetically diverse. They are different from the parent cell. This is the whole process of meiosis. You're going to notice some similarities between mitosis and meiosis. So both are going to start with interphase. There's going to be a period of DNA replication during S phase of interphase. And then we jump into PMAP, prophase, metaphase, anaphase, telophase. And then in the very end of meiosis 1, you have two daughter cells. Our two daughter cells are going to be genetically different from the original parent cell. But one of the things you'll see here is we're going to go through PMAP again. So prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis, we're going to get 4 haploid cells. So just a reminder, haploid means half the number of chromosomes that we originally had with the parent cell. You can see that here. Each of these cells is genetically diverse. They are different from the parent cell. And we're going to come back and talk a little bit more about this in our genetics review too. All right, now that ends our sales review. Feel free to go back and grab anything that you might have missed or re-listen if you want to. I hope that was helpful, and I will see you in the next review.